This is part 2 of 7.1. So we're going to have a quick review again of the work that led to the discovery of DNA structure. So what was the work? Well, there were two major things. One was the work by Chargaff, and this suggested a particular ratio. There was a particular ratio found within DNA where A was always at the same ratio or the same percentage as T, and G was at the same percentage as C. And so this was very strong evidence that these paired together. So adenine and thymine, guanine and cytosine. And the other thing that they had was the X-ray crystallography. They had this picture that suggested this helical shape, and it also, from working out the sizes, they knew how far apart the rungs of the ladder were. And then they also had the composition of the bases, and they knew the structure well enough to think about where bonding might happen. So, yeah, that's just a quick summary then. Chargaff's ratio, C and G having the same, A and T having the same. X-ray crystallography, by sending X-rays through. DNA doesn't actually crystallize very well. It doesn't really form crystals, but it was good enough that they could get the shape of it, because it doesn't form regular repeating crystals, but it was using the same technique. And so this was also diffraction, because as they go through, they kind of spread out. So the diffraction caused the X-ray film to develop. And here was that picture which was produced from Rosalind Franklin's work, where you can see these kind of repeating units. You see the helical shape, and you can also work out how many nanometers apart the rungs of these ladders were. And so that's what Watson and Crick then used. So who were Watson and Crick? What did they discover? And what did their work suggest in terms of DNA replication? Well, here they are. They were two scientists who were working, hoping to discover the structure of DNA. And they were working with cardboard cutouts, using modelling in order to try and develop their structure. And they used the X-ray crystallography picture, and they used Chargaff's ratio. So in their model, they had sugar and phosphate as the backbone, with nitrogenous bases pointing inwards, and this matched their evidence. And they could also see how hydrogen bonds would link the nitrogenous bases from the two strands. Now this suggested, because it was hydrogen bonds between the two strands, it suggested semi-conservative replication, and we'll come to that. It's where if we have two parent strands, when we copy DNA, each new strand, because this is double-stranded, we need to have two double-stranded. Well, each new will have one which was from the parent and one which is newly synthesized. And that means it's semi, half conservative. Conserve means to keep, so it's half keeping. It's keeping half the original, and it's adding one new strand in. And so their model, because they could see that it was down the middle, it was hydrogen bonds holding the two strands together, which are quite weak, but it was covalent bonds along the strands. That means that then it would be easy to pull these two strands apart and to use one as a template. So our next question is about the structure of DNA, and how is DNA structured? Well, it's structured around nucleosomes. So in eukaryotes, in species that have a nucleus, we have packaging of DNA. And it's packaged up we have DNA associated with protein. So we're using protein to help package. And these proteins are histone proteins. So if we think about the fact that it's not just DNA, it's DNA and protein, we can call DNA and protein chromatin. It's DNA wound around proteins. So if you think about if you had a very long piece of string, the way that we keep string packaged is that we wind it around a reel. So we can get lots and lots of string around a reel in a very condensed way rather than just having all of the string by itself. And so a nucleosome is the version of the reel. It's a protein reel around which we're going to wrap the DNA. So here's our nucleosome structure, and you can see that we've got 1, 2, 3, 4, 5, 6, and behind it's the same thing. It's like this. We've got basically 8 balls. And so these are 8 histone proteins. So there are 8 histone proteins which come together to act like the reel to wrap around the DNA. And we've got this other protein here, this linker, which is holding the DNA on. And so a nucleosome is made out of these 2 different types of histone, one which makes up the 8 balls, and because it's 8 we can say it's an octamer. So an octamer of 8 histone proteins and 1 linker protein. And then you can see that the DNA is wrapped around, and it's almost 2 turns. It's going around almost 2 turns. It's actually exactly 1.7 turns, which is 146 bases of DNA. Wraps around, and then it goes off, and then there'll be another nucleosome. So let's just have a look at all that. So a whole nucleosome is made out of DNA. It's made out of histone proteins, and there's an octamer. So the whole thing, the nucleosome structure, is core DNA. So we have DNA wrapped around an octamer of 8 histone proteins with a histone H1 which is a linker and holds the DNA onto those other histone proteins. And so what this creates is it creates a fibre that is wider. And the whole diameter of this is 30 nanometers. So this is what's called the 30 nanometer fibre. So if we zoom in here, we see it. We get DNA wrapped around those 8 histone proteins with a linker. And then we get them wrapping up, and we get overall, you can see it's kind of like a layer of 2. We get overall beads on a string, and those beads on a string, what's called the beads on a string of the nucleosomes, then come together to make what's called the 30 nanometer fibre, or the 30 nanometer solenoid, you will see. So this whole thing here, that is 30 nanometers. And then what that does is that then goes backwards and forwards and backwards and forwards and backwards and forwards, and it creates an extended form of the chromosome. And so we see that this kind of goes backwards and forwards and then is arranged and arranged and kind of wrapped around until eventually we get to the X-shaped structure. Now, it's important to say that these structures are only ever formed when we are copying DNA. Normally, when we're not copying DNA, we would have the kind of beads on a string structure of DNA. It wouldn't be wrapped up into these tight structures, these X shape. So only during DNA replication do we get this structure being shown, and we're packaging it up. So that's one of the functions of nucleosomes. It's to help package for copying DNA. Okay, see if you can stop the video and think about what were all of these different structures made from. So we had, as a quick review, we had DNA, then wrapping around our 8 histone proteins to make the beads on a string. Those coming together to form this 30 nanometer fiber, which then go backwards and forwards and backwards and forwards, and then we get this section of what we call chromatin, which is the DNA and protein, until eventually we get the whole chromosome form.